All right, let's go.
Number 12. The warm little pond. Darwin's idea that started everything. In 1871, Darwin wrote a private letter to his botanist friend Joseph Hooker. In it, he speculated, quietly and without publishing it, that life might have begun in a warm little pond, a shallow body of water rich in ammonia, phosphorus salts, light, heat, and electricity, a chemical soup energized by lightning, something that could produce a protein compound ready to undergo still more complex changes. He never published this idea. He thought the world was not ready. 82 years later, Stanley Miller and Harold Urey tested it. In 1953, Miller filled a sealed glass apparatus with methane, ammonia, hydrogen, and water vapor, the gases believed to represent the early Earth atmosphere. He ran electrical sparks through the mixture continuously for a week, simulating lightning strikes into a primordial ocean. The results stopped the scientific world. Within days, the apparatus produced amino acids, 5 of them, including glycine and alanine, 2 of the most fundamental building blocks of proteins found in every living thing on Earth. Chemistry alone. No biology, no cells, just electricity and simple gases and a week of time. The Miller-Urey experiment proved that the chemistry of life was not miraculous. It was inevitable given the right conditions. The building blocks of proteins form spontaneously from the ingredients available on the early Earth. But amino acids are not life. They are letters. Life requires words, sentences, a library. The Miller-Urey experiment showed the letters could appear, how they assembled into the first self-replicating molecule, the first thing that could copy itself and pass information to its copies, remains the central unsolved problem. Darwin's warm little pond is still the starting point, but the story of what happened inside it is still being written. Number 11. The RNA World, the molecule that did everything. Here is the problem with the origin of life that took decades to even properly state. Life requires 2 things simultaneously: a molecule that carries information, a blueprint, and a molecule that reads that blueprint and builds things. In modern life, DNA carries the information and proteins do the building. But proteins are built using instructions from DNA, and DNA is copied using proteins. Each one requires the other to exist. Which came first, the chicken or the egg, does not begin to capture how circular this problem is. The RNA World hypothesis, developed through the 1960s and formalized in the 1980s by Walter Gilbert, proposes a solution. Before DNA, before proteins, there was RNA. RNA is extraordinary. It can carry genetic information like DNA, but it can also fold into 3-dimensional shapes and catalyze chemical reactions like a protein. One molecule doing both jobs simultaneously. A chicken that is also an egg. In 1982, Thomas Seck discovered that RNA could catalyze its own chemical reactions, RNA molecules that act as enzymes. He called them ribosomes. It won him the Nobel Prize in 1989. If RNA can carry information and catalyze reactions, including potentially the reaction of copying itself, then a single RNA molecule arising by chance in the right chemical environment could be the first self-replicating entity, the first thing that made more of itself, the first ancestor of every living thing on Earth. The RNA World hypothesis is the closest thing science has to a consensus answer for the origin of life. It is taught in universities. It is the framework most origin of life researchers work within. It is also full of unsolved problems. The spontaneous formation of RNA from simple chemicals has never been fully demonstrated in the laboratory. RNA is a complex molecule. Getting its components to assemble in the right sequence without the cellular machinery that normally does this assembling is an unsolved challenge. The conditions under which early Earth RNA could have formed, the concentrations required, the mineral surfaces needed, the energy sources involved, are still actively debated. The leading theory, still not proven, still being worked on. In 2026. Number 10. The hydrothermal vent theory. Life in the dark. For most of the 20th century, scientists assumed life needed sunlight, energy from above, the warm pond, the lightning, the surface chemistry. In 1977, a team of oceanographers in the submersible Alvin descended to the Galapagos Rift, a volcanic spreading zone on the Pacific Ocean floor, and found something nobody expected. Life. Abundant, extraordinary life. Tube worms 2 meters long, clams the size of dinner plates, fish, crabs, an entire ecosystem thriving in complete darkness at depths where the pressure would crush an unprotected human in seconds. The energy source was not sunlight. It was chemistry. Hydrogen sulfide pouring from hydrothermal vents reacting with the surrounding seawater. Bacteria using this chemical energy through a process called chemosynthesis. Everything above them in the food chain feeding on the bacteria. Life did not need the sun. Life needed chemistry. The alkaline hydrothermal vent theory, developed most thoroughly by Michael Russell, goes further. Alkaline vents, like the Lost City hydrothermal field in the Atlantic Ocean, produce not scalding acid water, but warm alkaline fluid through a process called serpentinization. These vents create natural proton gradients across thin mineral membranes. Here's why that matters. Every single living cell on Earth runs on proton gradients. The inner membrane of the mitochondria, the power plant of every cell in your body, works by pumping protons across a membrane and using the resulting gradient to generate ATP, the universal energy currency of life. The chemistry of alkaline hydrothermal vents naturally produce proton gradients across mineral surfaces, the same driving force that powers every cell on Earth, present in rocks on the early ocean floor, with no biology required. Life may not have started on the sunlit surface at all. It may have started in the permanent darkness of the deep ocean, where chemistry provided everything biology would eventually internalize and make its own. Number 9. Metabolism First. Life without a blueprint. Every theory discussed so far starts with a molecule that replicates, something that copies itself and passes information to its copies. Replication first, information first, blueprint first. Gunther Wachter proposed something completely different. What if life did not start with replication? What if it started with metabolism? Metabolism is the network of chemical reactions that captures energy and produces useful molecules. Every living cell runs metabolic cycles, the citric acid cycle, the Calvin cycle, chains of reactions that transform simple molecules into complex chemistry of life. These cycles require enzymes, protein catalysts to run. But Wachter proposed that on the mineral surfaces of iron-sulfur compounds on the early ocean floor, the same cycles could run without enzymes. The mineral surface acts as the catalyst. The reactions are driven by chemical energy available in the environment. A metabolic cycle running on a mineral surface is not alive. It does not replicate. It does not have a genome. But it captures energy, it produces complex molecules, it creates local pockets of chemistry that are far from equilibrium with the surrounding environment. Wachter proposed replication came later, after metabolism was already running on mineral surfaces, building complex molecules continuously. The conditions arose for information-carrying molecules to appear, not as the starting point, but as a later development in a system that was already doing chemistry. Metabolism first versus replication first is one of the deepest divisions in origin of life research. Both camps have serious scientists, both have serious experimental support. Neither has won. The argument has been running since the 1980s. It is still running in 2026. Number 8. The Lipid World. The bag that made itself. Before replication, before metabolism, before any chemistry at all. A bag. Fatty acid molecules are simple. They have a water-loving head and a water-heating tail. Drop them in water and they spontaneously arrange themselves into hollow spheres, bilayer membranes with water inside and water outside. No chemistry required, no blueprint required, just physics. These protocells can grow by incorporating more fatty acid molecules from the surrounding water. They can divide when they get large enough, physical forces pulling them apart into daughter vesicles. They can encapsulate other molecules that happen to be present in the surrounding water, trapping useful chemistry inside. Jack Szostak at Harvard has spent decades showing that these protocells are remarkably capable. They can take up nucleotides, the building blocks of RNA, from the outside and concentrate them inside. Under the right conditions, nucleotides inside protocells can assemble into short RNA strands, RNA that can copy itself. The Lipid World proposes that the membrane came first, not as a sophisticated cell membrane requiring complex protein machinery to build, as a spontaneous physical consequence of fatty acid chemistry in water. The bag assembled itself, and then chemistry happened inside. The first cell may not have been the product of a long chain of molecular evolution. It may have been a spontaneous physical structure that captured chemistry by accident and found that chemistry useful. Number 7. The clay hypothesis. Life on stone. Alexander Graham Cairns-Smith was a Scottish chemist who in 1966 proposed something so radical that it was largely ignored for decades. Life did not begin in water. It began on clay. Clay minerals are crystalline structures that form naturally in water, and crystals have a remarkable property. They replicate their structure. As a crystal grows, it incorporates surrounding molecules and forces them into its own pattern. The crystal structure is self-perpetuating. Defects in the crystal, impurities and irregularities, are copied as the crystal grows. Information stored in defect patterns is passed from parent crystal to daughter crystal. Cairns-Smith's hypothesis is the clay crystals were the original genetic material, not RNA, not DNA, not proteins, crystal lattice defects in clay minerals. The first heredity was geological. Organic molecules, amino acids, nucleotides, could have assembled on clay surfaces using the crystal structure as a template. Over time, the organic chemistry became more sophisticated. Eventually, the organic molecules became capable of replication on their own, and the clay scaffold became redundant. Life abandoned its mineral origin and took its chemistry into solution. The idea is called genetic takeover. Inorganic crystal genetics replaced by organic molecular genetics. Cairns-Smith's hypothesis is not mainstream. It has never achieved the experimental support of RNA World, but it has never been disproven either. And it solves a genuine problem. It provides a plausible mechanism for heredity before RNA, without requiring the spontaneous appearance of a complex self-replicating molecule. Clay as the original genome, it is either completely wrong or one of the most profound ideas in the history of science. We do not know which. Number 6. The Formamide Theory. The simplest molecule that builds everything. Formamide. 1 carbon, 1 hydrogen, 1 nitrogen, 1 oxygen. One of the simplest organic molecules that exists. It is also found throughout the universe, in molecular clouds, in cometary material, in the interstellar medium. Wherever organic chemistry happens in space, formamide is there. And in 2001, researchers discovered something extraordinary about it. When formamide is heated by volcanic activity, by meteorite impacts, by ultraviolet radiation, it spontaneously produces all 4 nucleobases of RNA and DNA: adenine, guanine, cytosine, uracil, the chemical letters of the genetic code. From one simple molecule, without requiring any complex starting chemistry. The Formamide Theory proposes that the early Earth, bombarded by meteorites and volcanically active, had concentrations of formamide in evaporating pools and hydrothermal environments. The energy available from impacts and volcanism drove formamide reactions that spontaneously produced the building blocks of RNA and DNA. Not a long, complex chain of chemical steps, one molecule, one energy source, all 4 genetic letters. The simplicity of this pathway is its strength. The origin of life does not require an improbable sequence of chemical accidents. It requires formamide and energy, both abundantly available on the early Earth and throughout the universe. Number 5. Panspermia. Life did not start here. What if the question is wrong? What if life did not originate on Earth at all? Panspermia, from the Greek for seeds everywhere, proposes that life or its chemical precursors are distributed throughout the universe and can travel between planets on asteroids, comets, and interplanetary dust. The idea has serious scientific support. Bacteria have been shown to survive the vacuum of space, the radiation environment of interplanetary space, and the heat of atmospheric entry when protected inside rocks. The Chelyabinsk meteorite that exploded over Russia in 2013 was found to contain amino acids. The Murchison meteorite that fell in Australia in 1969 contains over 90 amino acids, 19 of which are found in living organisms on Earth. JAXA's Hayabusa 2 mission returned samples from asteroid Ryugu in 2020. The samples contained amino acids in uracil, one of the 4 genetic letters of RNA. The chemistry of life is not confined to Earth. It is present in space, in asteroids, in comets, in the interstellar medium. Panspermia does not necessarily propose that fully formed life traveled from another star. It can operate at a simpler level. Chemical precursors seeding planets with the building blocks from which life can assemble locally, or it can operate at the most extreme level, microorganisms surviving interstellar travel inside rocks ejected from one planetary system to another over millions of years. What panspermia cannot do is explain the ultimate origin. If life came to Earth from Mars, where did Martian life originate? If it came from another star system, where did it originate there? Panspermia moves the question. It does not answer it. But if life originated elsewhere and arrived here, the timeline of Earth's history makes more sense. Life appeared on Earth astonishingly quickly after the planet cooled enough to be habitable, almost too quickly, as if it was already waiting to arrive. Number 4. Directed panspermia. We were planted here. In 1973, Francis Crick, the man who co-discovered the structure of DNA, published a paper proposing something that would have seemed insane from almost anyone else. Life on Earth was deliberately seeded by an extraterrestrial civilization. Crick called it directed panspermia, not accidental delivery by asteroid, intentional seeding, a spacecraft carrying microorganisms sent from another civilization in another star system to Earth, and possibly to other planets, billions of years ago. Crick's argument was specific. The genetic code, the system by which DNA triplets code for specific amino acids, is universal across all life on Earth. Every bacterium, every plant, every animal uses the same code. It is so universal and so specific that Crick argued it shows signs of having been optimized, not the messy result of random chemical evolution, but something that looks selected, engineered. He also noted the frozen accident problem. The genetic code could have been different. Many alternative codes would work, but every life form on Earth uses the same one, as if all life descended from a single small founding population, a founding population small enough to fit on a spacecraft. Directed panspermia is not accepted by mainstream science. It requires a civilization advanced enough to interstellar travel to have existed billions of years ago, long before Earth formed. It pushes the origin of life question entirely outside of Earth and outside of the observable history of the solar system. But Francis Crick was not a fool. He was one of the most rigorous scientists of the 20th century. He proposed it not as mysticism, but as a scientific hypothesis that fits the available data at least as well as some of the alternatives. The question he was really asking was this: is the origin of life so improbable that it could only have happened once or a very small number of times in the history of the universe? And if so, are we the descendants of that one origin? We do not know. Number 3. The electric spark in ice. Life in the cold. The dominant assumption in origin of life research is that life requires warmth. Hot vents, warm ponds, volcanic environments where chemistry is energetic and molecules are mobile. Christoph Biebercher and others have proposed the opposite: ice. Frozen environments do something chemically remarkable. They concentrate. As water freezes, it excludes dissolved molecules into the remaining liquid channels between ice crystals. A molecule present at 1 part per billion in open water can reach concentrations of 1 part per thousand in the liquid channels of sea ice, a concentration increase of 1,000,000-fold. At those concentrations, chemical reactions that would never occur in dilute solution become possible. RNA precursors concentrated in ice channels can assemble into short RNA strands. The cold slows degradation, protecting fragile molecules that would rapidly break down in warm water. UV radiation still penetrates shallow ice, providing energy without the destructive heat. The early Earth went through periods of global glaciation, snowball Earth events where ice covered much of the planet's surface. These events are usually considered catastrophic for life. The ice origin hypothesis flips this. The glaciations were not obstacles, they were incubators. Life may have begun not in warmth, but in cold, not in activity, but in stillness, in the quiet chemistry of frozen channels, where molecules were brought together by the physics of freezing and given the time and concentration to do something extraordinary. Number 2. The simulation hypothesis. The question behind the question. This one sits at the edge of what can be called science, but it has been taken seriously by serious people including Nick Bostrom, Elon Musk, and a significant fraction of physicists and philosophers, so it belongs on this list. The simulation hypothesis proposes that the universe, including Earth and all life on it, is a computational simulation running on the hardware of a more fundamental reality. If this is true, the question of how life began on Earth is not a chemistry question, it is a programming question. Life appeared on Earth because the simulation was designed or initialized in a way that produced life. The biochemistry of RNA and proteins and membranes is not the mechanism of life's origin. It is the rendered output of a computational process running at a deeper level. Nick Bostrom's 2003 simulation argument makes a specific probabilistic case. One of 3 things must be true. Almost all civilizations go extinct before developing the ability to run realistic simulations of consciousness. Almost all civilizations capable of running such simulations choose not to, or we are almost certainly living in a simulation right now. The logic is tight. If even one civilization in the universe's history has developed the ability to run realistic consciousness simulations and has chosen to do so, the number of simulated minds would vastly outnumber the biological ones. The probability of being a biological original rather than a simulated copy would be vanishingly small. If we are in a simulation, the origin of life is not a mystery to be solved by chemistry. It is a design decision made by whoever wrote the initial conditions. The deepest problem with the simulation hypothesis is also its most interesting feature. It is almost impossible to falsify. Every observation we could make is consistent with a sufficiently sophisticated simulation. Every physical law, every chemical reaction, every quantum event could be simulated output rather than fundamental reality. Which means, if it is true, we may never know. Number 1. We do not know. The most honest answer in science. Here is the truth that every other entry on this list is trying to work around. We do not know how life began on Earth. Not approximately, not almost, genuinely, completely do not know. We have been to the moon. We've landed spacecraft on asteroids and comets and Mars. We have mapped the human genome. We have detected gravitational waves from black holes merging 1 billion light-years away. We have imaged a black hole directly. We have built quantum computers and gene editing tools and particle accelerators that probe the structure of matter at scales 1 billion times smaller than an atom. We cannot explain how the first living thing appeared on the planet we live on. Life emerged on Earth approximately 3.5 to 4 billion years ago, possibly earlier. Some evidence suggests microbial life as early as 4.1 billion years ago. Earth itself is 4.54 billion years old. Life appeared within the first 400 to 500 million years of the planet's existence, cosmically fast. Every theory on this list has evidence supporting it. Every theory on this list has serious unsolved problems undermining it. No single theory has achieved scientific consensus. No single experiment has demonstrated the complete pathway from simple chemistry to the first self-replicating entity. The RNA world is the closest thing to a leading answer. It has the most experimental support, the most laboratory demonstration, and the most active research effort behind it. But it has not been proven. The spontaneous formation of RNA under early Earth conditions has never been fully demonstrated in the laboratory. What we have is a collection of plausible stories, each one grounded in real chemistry, real physics, real geology. Each one consistent with some of the evidence, none of them the full answer. And here's what makes this the most important unsolved problem in science. If we understood how life began on Earth, we would know whether life is common or rare in the universe. If life emerges inevitably given the right conditions, the universe is teeming. If life requires an improbable sequence of events, we may be alone in an unimaginably vast cosmos. The answer to how life began here is the answer to whether anyone else is out there. We have been asking that question since the first human looked up at the night sky and wondered. We are still asking it. 12 theories about how life first appeared on Earth. From Darwin's warm little pond to RNA molecules that copy themselves. From chemistry in the dark of the ocean floor to crystals on ancient rock. From simple molecules found throughout the universe to microorganisms traveling between stars. From a deliberate act of seeding by an ancient civilization to the possibility that chemistry itself is a simulation. None of them proven, all of them real science. All of them pointing at the same gap in human knowledge, the gap between chemistry and the first thing that was alive. We came from somewhere. We do not yet know where. Subscribe to Cosmic Hives. See you in the next one.